Hello everyone, now welcome to the last module, that is module 3.3 of MOOC on Bioinformatics Essentials. This module will be based on multiple sequence alignment. In this module, we'll learn about how to means what is the theory of multiple sequence alignment, why multiple sequence alignment is required, what are the fundamentals of this, and everything that is required to uh, proceed before the multiple sequence alignment. So we'll start without wasting time. See, why compare more than two sequences? Okay, so pairwise alignment in module 3.1 part 2, or I guess module 3.2, we have learned about the pairwise alignment. So why to compare two sequences, that is elaborated over there. So pairwise alignment compares two sequences, but biological research often involves multiple species, protein families, gene families, related organisms, multiple variants of protein, and researchers may want to identify conserved residues, okay, or variable regions, mutations, evolutionary patterns, functionally important positions. So, where we will compare a same gene, a same protein sequence from various species, and we'll find what are the conserved domains or what are the variable regions, if there is any mutation, at which position there is a mutation, what are the evolutionary patterns, that thing will observe using the multiple sequence alignment. Now, what is a multiple sequence alignment? What is the actual meaning of multiple sequence alignment? Multiple sequence alignment arranges three or more related biological sequences so that corresponding positions can be compared. Okay. So in this process, we arrange three or more related biological sequences. Okay. And then we do the comparison between them. Now, for example, you can see here human, one example sequence is given MKTLLAAGT. This is a protein sequence. Mouse MKTLLAAGC, and rat MKTLLAAGC. And you can see here the third position in human is last position in human is different, and in mouse and rat it's a different. So this alignment allows researchers to identify position that remain conserved across sequence. Means, see here, this MKTLLAAG, this position is common throughout the sequence. So this is the benefit, or actually this thing we do in the multiple sequence alignment. Now, why these conserved positions are important? If position remains unchanged across many related proteins, that position may have biological importance. If any position or any sequence, any bit or any chunk of sequence is common throughout the species, that means that chunk of or that byte of sequence is important. This will have some biological significance. How? This may contribute to catalytic activity, they may contribute to structural stability, they may contribute to binding, ligand binding, or they may contribute to protein interaction. So, however, conservation alone does not prove the function. It provides a clue that can be investigated further. So, if there is a thing, if there is the conserved position, or if there is any conserved chunk of the sequence, that doesn't mean that it's a function. We can find the function using the ontology or further study. But yes, we will get a clue that yes, there is a something between this species, or this thing is biological significant. Now, everyday example, how we can correlate with our everyday thing. So imagine a photograph of members of same family. Okay, your close relatives. Consider now. Certain features may remain similar: eye shapes, nose structure, facial proportions. If you look at all the things closely, you will see eye shape, nose structure, facial proportions. Even you may notice some small things like voice and the height, etc., etc. But some characters may vary: hairstyle, clothing, hair color, etc. Some may have different clothing choices, like that. Okay, so multiple sequence alignment works similarly. It compares all the things and it gives you the picture that these things is common, these things are different throughout the sequences, and it builds the actual roadmap of how evolution is taken place between the concerned species. So it helps to distinguish conserved features from variable features, which features are conserved and which features are variable across related biological sequence. It's a key takeaway from this one. And cluster omega, now we'll do the demonstration on the cluster omega. Cluster omega is widely used tool for multiple sequence alignment. It's a free tool. It is created and it is hosted by the EMBL, European League of that Bioinformatics. So it is conserved by them. Sorry, it is handled by them. It can align protein sequences, RDNA sequences, RNA sequences. That will everything we'll see in the demo. And the resulting alignments, we can use that results to visualize conserved and variable positions. So till then, thank you for watching this theory video on the multiple sequence alignment. In the next video, we'll do the demonstration of cluster omega, and we'll do the multiple sequence alignment on the same. Thank you. Please give the like, share and subscribe to this video.